Hi and welcome back. It's a long time since we met. Today, different from our usual discussions or lectures on topics in the syllabus of anatomy and pathology, in this video, I will be discussing a hot current affairs topic. The hot discussion among the medical fraternity and general population alike, the buzz of the town, the new entrants in the town, the vaccines for SARS novel coronavirus 2019. We will discuss the process behind the manufacture of these vaccines, their mechanism of action, and relative merits and demerits in this video with the help of simple 3D animations done by me. As you might be knowing, our government has approved two vaccines for SARS coronavirus, Covishield and Covaxin. These vaccines are currently being administered to healthcare professionals and are expected to hit the general market anytime soon. First, we will see the science behind the Covishield vaccine, which is being actively administered and is considered to be a breakthrough and advanced technology in the field of vaccination. Covishield is a recombinant DNA viral vector vaccine. Here, DNA material of an adenovirus, which is a common resident of chimpanzees, is used as a vector to carry the gene encoding for the now famous spike protein of COVID-19. What is the science behind this? We will see that now. The scientists, what they have done in this case is, they have isolated the RNA from SARS coronavirus. As you might be already aware, it is an RNA virus. That RNA material was isolated. They specifically separated the region of the RNA which encodes for the spike protein. They then converted this RNA into DNA by adding a matching duplicate strand to make it a double-stranded double helix DNA. They found a suitable vector candidate to carry this gene into human cell in the adenovirus, which causes common cold in our ancestors, chimpanzees. They got hold of that adenovirus and extracted its DNA. Yes, this adenovirus is a DNA virus. So they made this DNA non-replicating and non-infective by removing the unnecessary parts, and then they attached the RNA of the coronavirus, now transformed into DNA as we saw already. They attached this DNA which encodes for spike protein to this, and this adenovirus with the modified genetic material, that is modified genome, is what is injected as Covishield vaccine. Once in the human body, this adenovirus enters the human cell and within the cell it is dismantled and the DNA enters the nucleus. Within the nucleus, this modified DNA using our own, that is the human cell's own inbuilt mechanism of protein synthesis, it synthesizes the coronavirus spike protein. Getting into its detail, this process involves two things, transcription and translation. The first one, transcription, happens within the nucleus. Here, the DNA region encoding for spike protein is transcribed into mRNA or messenger RNA. Messenger RNA is the RNA which carries the message in DNA to the protein making machinery of the cell. That's why it is called messenger RNA. It carries the message from DNA to the protein making machinery. This messenger RNA leaves the nucleus and reaches the cytoplasm. Here in the cytoplasm, the ribosome, which is the protein factory of the cell, attaches to it. As the ribosome moves along the mRNA, reading the genetic code in it, the tRNA or transfer RNAs bring matching amino acids to it. As the genetic code is read, matching amino acids for that genetic code is brought by transfer RNAs or tRNAs. Amino acids are the basic constituents of any protein. These amino acids are linked together and three-dimensionally folded to form the famous spike protein. This spike protein, so produced in our cell, leaves the cell and reaches the circulation. Here, they are challenged by our defense cells, that is WBCs, a special type of defense cells called macrophages. The macrophages eat or phagocytose that spike protein and present the antigen of the spike protein to another group of defense cells called B cells. These B cells get activated on this contact with the antigen, and then the activated B cells actively divide and start producing antibodies. These antibodies act against the spike protein, that is, in effect, they are antibodies acting against the SARS coronavirus itself. So, whenever there is a subsequent infection of SARS coronavirus, the virus is immediately challenged and destroyed by these circulating antibodies. And this is the story of Covishield. Coming to Covaxin, here the principle used is less complex, straightforward, rather primitive, but time-tested. Here, the SARS coronavirus, which is isolated, is killed or inactivated using certain chemicals like beta-propiolactone or formaldehyde, and this killed virus is what is injected into us. This killed or inactivated virus, though cannot cause active infection, can still elicit an immune response. And this immune response acts against subsequent infection. This is the principle behind this vaccine. This killed virus particle, just like the spike protein in the previous case, is challenged and phagocytosed by macrophage, and then the antigen is presented to B cells. B cells produce antibodies against this antigen, and these antibodies protect us against subsequent viral infection. Simple and straightforward. Now, coming to the relative merits and demerits of both. The technology behind Covishield is pretty advanced, but relatively untested in humans. There are so many studies of this on Ebola, HIV, etc., but none conclusive. So naturally, we don't know the long-term effects of this adenoviral vector DNA material inside us. It is said to be non-replicating and safe, but technology is new and not time-tested. There is no way to be sure it is safe. Again, no way to be sure that it is dangerous either. So a gray area there. Coming to the efficacy, the data available is variable. A half-dose, full-dose study has given a 90% efficacy. Efficacy in other studies range from 60 to 90%. Here, I must also add one thing. This vaccine elicits both B cell and T cell mediated immunity, and hence it should be more effective, and the effect or the protection should last longer. Despite the gray areas, given that this technology is pretty advanced and very effective, and the strong government backing and wisdom of experts behind the decision, my opinion is it should be safe and effective. But do keep in mind the vaccination eligibility has some inclusion and exclusion criteria like bleeding disorders, immunocompromised, etc. Watch for it if you have any of these. Otherwise, it is pretty safe. As far as Covaxin is concerned, the technology is primitive but time tested. It is a technology used in various human vaccines like rabies, etc. But the inherent problem with kill vaccines is they are less effective and the effect is short lived and need booster doses. And another issue specific to Covaxin here is it has been approved in an emergency clinical trial mode. That means the trial studies of it are not yet complete. Still, since this technology itself is time tested and very commonly used by us already for so many years, even though the trial for this particular vaccine is incomplete, it should be safe. So no problem there either. Now that we have discussed the mechanism behind and the relative merits and demerits of both, I think we will be able to go for the vaccination more confidently. And that's all for now, and until we meet in another video, thanks for watching. Bye.